Okay, assalamu alaikum everyone. My name is Yazan Almasry. I'm an intern, and today I'm going to be teaching you hormones 1 and 2. So I've looked through these lectures. They're a little bit of like basics. So I'm going to be trying to cover the basics as much as possible and try to make it as easy as possible. And at the same time, I'm going to like be mainly covering only the relevant information. So things that I didn't include in the lecture, they're not that important, honestly. And I basically try to include everything. So hopefully I cover everything. If you have any question, if there's anything that's not clear, don't wait until a break. Just stop me immediately. And that's either by sending in the chat or just unmuting and saying so. All right. Okay. So this lecture is about hormones. And we probably all have heard about the word hormones in our lives. Okay. Now, here's the thing. Does anyone know what are hormones? Can anyone like take a guess what are hormones? Chemical messengers, chemical. Exactly. So basically, in our body, okay, we have one of two methods of communication between cells. We have so many cells. We have trillions of cells, hundreds of tissues, and many organs that are doing different job. But we need something to make them work together, something to connect them. And this is going to happen by one of two things. Okay? Before we get into that, I'll give some examples. So, I don't know if you guys heard this before, but some people mention like whenever they eat, their insulin rises. Like they're like, oh, I don't want to eat like a snack, so my insulin does not spike, right? Or during stress, cortisol level will spike, for example, will rise. Have you guys heard of these before or no? Okay, yes, perfect. So these are some examples of hormones, and basically they're basically a response to stress in the body. Okay, and we're gonna talk about further in the next slides. So for the cells to work together and to bring the body together, we need two major things, two major system. Okay, the central nervous system or the nervous system in general, and the endocrine system. And you guys have already covered the nervous system, and you covered how the electrical impulses between neurons basically cause the interconnection uh, and the action as a reaction to whatever is external to the body. For example, if you touch a hot stove and you immediately move your hand, that's basically done by the nervous system. Now, on the other hand, we have something called the endocrine system. Okay? And what the endocrine system basically does, it's relying on these chemical messengers called hormone. Okay? Now, the difference between the nervous system and the endocrine system is that the endocrine system is a slower version of the nervous system. Okay? As it's slower, it also has a longer lasting effect. All right? So I'm going to repeat myself. We have two major systems that control the body's actions. Okay? That controls whether a human being grow, a human being responds to a certain thing. These two systems are a nervous system and the endocrine system. All right. And our focus for today is the endocrine system. Okay. So it is important to know that the endocrine system relies on something called hormones, okay, which are also chemical messengers. And it's important to know that these hormones are slower onset compared to nervous system and last longer. Okay. Also, it's a relative term. When we say slower, we're talking slower than neurons. Okay, so it's not as fast as moving your hand from a hot stove. Okay, but it does not necessarily mean it's slow, like it's going to take months. It take days, sometimes minutes, sometimes months. So it really depends on the type of hormone. All right, so this is our focus for today, the endocrine system. Okay, so what exactly is a hormone? Okay, and we said hormone is a chemical messenger that is released by an endocrine gland into the bloodstream to regulate the functions of target cells. Okay? Now, I want you to forget about the word distant because it can be close, but this is just the definition. Okay? Now, it's important to know these characteristics. You don't have to memorize them, just understand them. Okay? Hormones are released in very small amounts. Okay? And the reason for this is that because you only need a small amount to produce a huge action. So there is no need to release a lot of it. Okay? Hormones will travel through the blood and they affect specific cells. So they're not acting haphazardly. Okay? And the reason behind that is because they have specific receptors they bind to. All right? Does that make sense? Mm, 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 mm. Yeah, okay. So again, hormones, what's their main function? The chemical messenger, their main function is to regulate cellular activity. Okay? Now they do not come up with new, sorry, they do not come up with new reaction. They just regulate already existing ones. Okay. These are just some concepts, okay? You don't have to memorize any of the thing that I'm saying. Just try to understand it. All right. Now, there are different types of cell signaling, okay? And the type of cell signaling here is going to depend on how it's being done, okay? So what I mean by this, we have different names here. In crin or crine basically refers to like something related to hormones, okay? So all of these are related to hormones, the ones that have crine or crin in it, okay? So when we say autocrine, which is right here, crine is what it okay. So autocrine is basically, or autocrine, it's basically auto means on itself. So basically this cell is going to release hormones, and these hormones are going to act on the cell itself. Okay. Now we have something else. So this is the first type. The second type is paracrine or paracrine. Paracrine, what it's going to do is basically we're going to have a cell that's releasing hormones. Now these hormones are going to go to a cell that's close by. So it has to be close. And then binds to the cell that's close. Okay? So the difference between autocrine and paracrine is basically autocrine, it's acting on itself, and paracrine, it's acting on close cell. Okay? So auto means on itself. Okay? Now we have endocrine, the third type. What does endocrine do? It basically releases it to the bloodstream, and then from the bloodstream, it leaves to act on cells that are far away. So this one is far. All right, so these are the three main types. And then we have neurotransmitters, which you've already covered, guys, which is basically release of neurotransmitters at the synapse of the neuron, like acetylcholine, norepinephrine, etc. Have you guys covered this? I think you did, right? Yeah, okay. And then we have something called neuroendocrine, which is essentially neurons. Okay, so we have a neuron, very ugly one. It's releasing hormones into the bloodstream, and then it will go to the cell and act on it. All right, so this is what it is, neuroendocrine. All right, are the five types clear? Go ahead. I think someone had a question or... All right, I guess not. Okay, play. So this whole thing is the endocrine system. All right. Now I want you to, let's take it like step by step, okay? The major part that controls all of this is two things. Number one is the hypothalamus. Number two is the pituitary gland. All right. Now, the hypothalamus, what it does, it's right here. It's the one that controls the pituitary gland. Oh my God, it's going crazy. It's the one that controls the pituitary gland, okay? And then the pituitary gland is going to release hormone for the rest of the body's glands. All right. Does that make sense? Now, so what releases hormones to the rest of the body and is known as the master gland is the pituitary gland. Now, what controls the pituitary gland is the hypothalamus. So the hypothalamus is in control of all, basically. If the hypothalamus goes wrong, that pituitary will go wrong, the rest of body's gland will go wrong. Okay? Same thing. If the pituitary gland goes wrong, the rest of the body's gland will go wrong anyways. Okay? Does that make sense? Now, in the body, we have something that's called a gland. Now, a gland is basically the gland's sole purpose, or a lot of the gland's sole purpose, is to release hormone. So, whenever we mention a gland, we're talking about releasing hormones. Okay? Now, it's important to know that hormones are also released by other organs. Essentially, all organs can release hormones. So, can someone give me an example of an organ? And I'll try to give you a hormone that's released by the organ. CCK. So CCK, I think, is released by the duodenum, right? I don't remember anymore. Okay, so the heart, for example, since you asked the heart releases something called AMP, atrial natriuretic peptide. Exactly. That's a hormone. And this hormone is going to cause vasoconstriction, I think, if I'm not wrong. Or vasodilation, actually. Vasodilation. Yeah. It causes vasodilation, inhibits the rest. You're right. Okay, so let's review all of what I said. So hypothalamus is the main control center of the endocrine system. And then we said the pituitary gland is the master gland. And we said the pituitary gland releases hormones that regulate other endocrine glands. Okay? Now, in the pituitary gland, or the type of or hormones that are released from the pituitary gland, what they do is they activate other glands. Okay, so it activates the thyroid gland, it activates the adrenal gland, it activates the gonads, and yeah, these are the main three. So any hormone that's released from the pituitary, right, that activates, let me write this down, activates other glands is known as a trophic hormone. So if you're asked what's a trophic hormone, it's a hormone that's released by the pituitary gland that activates other glands. Okay? And we have four trophic hormones. So what are these four? We have TSH, thyroid stimulating hormone, activating the thyroid. ACTH, adrenocortical thyroid, I forgot the full name, hormone. It basically activates the adrenal gland or adrenal cortex. Then we have, so this is number two. Then we have FSH and LH, which act on the gonads, which is either the testes or the ovaries in females. All right, is everything clear in this slide? Perfect. If there is anything that's unclear, feel free to ask. Don't feel shy about it. Okay. And then we mentioned this, that hormones are not only produced by glands, they're also produced by regular organs. And we said, for example, the heart, we have AMP. The kidneys, we have erythropoietin. Liver, we have insulin growth factor. And the list goes on. Okay. So basically, the endocrine system is a widely spread system. It's not only involving the endocrine glands. All right. So let's talk more about the trophic hormones. Okay. Now, I don't know if you guys had any lectures about the pituitary gland. Did you guys have any lectures about the pituitary gland? Oh, perfect. So you guys have heard about the anterior pituitary, posterior pituitary, and all of stuff? Okay, perfect. So essentially, this is the pituitary gland. We have two parts of it. We have anterior and posterior. Okay. In the anterior pituitary, it has a different name. It's called adenohypophysis. I cannot pronounce today. Adenohypophysis, I think. I wrote it somewhere, but I don't know where. And the posterior is neurohypophysis. Adenohypophysis and neurohypophysis. Exactly. So the way I remember it is because anterior with an A. So A for adeno. Anterior, you can just remember it's neuro. Okay? And the reason it's named this way, because adeno means gland, right? So the anterior pituitary acts like a gland. And it's basically releasing hormones. While the posterior pituitary What do you mean it's flipped? No, no, no. I think this yeah, you're right. I think I flipped the whole thing. Well, let's scratch that. All right. So, thank you for letting me know though. So, anterior pituitary and posterior pituitary. Actually, it's not flipped. I think it's the right direction, but I messed it up. So, anterior for adeno, which is the gland that's releasing hormones. Posterior for neuro, and the reason it's neuro because it's neurons that are coming all the way here and releasing the hormones, coming from the hypothalamus, releasing hormones. Okay, this is why they call it adeno and neuro hypophysis. Okay, so the adenohypophysis, or the anterior, is the one that releases the trophic hormones that we just talked about. Okay, and we mentioned the four trophic hormones and where they act on. Okay, so there are two main pathways from the hypothalamus to the pituitary gland that controls the pituitary gland. Okay, and I think you're going to take this even in a different lecture. Okay, so hypothalamus, we mentioned that the hypothalamus controls what? The pituitary gland. Okay, and we have two main pathways that controls it. We have the hypophyseal portal system, okay, which is this one, this portal system. And this basically is a venous system. And what happens is that at the median eminence, this area is called the median eminence, the hypothalamus is going to start releasing hormones here, okay? And these hormones are going to be transported through this portal system into the anterior hypothalamus, into the anterior pituitary, okay? Or the adenohypophysis, okay? Now, these hormones are going to bind to the adenohypophysis and activate the adenohypophysis or inhibit it, okay? And then, because that's why we said it controls it by these hormones. And then the pituitary gland can release it into the rest of the body. All right, let me repeat that again. So we have the hypothalamus here. It controls, right? So how does it control? At the median eminence, we have the hypothalamus releasing hormones into the portal system, the portal venous system. Okay? These hormones are going to diffuse down into the anterior pituitary gland or the adenohypophysis, activate or inhibit the adenohypophysis, which will cause the release of hormones or inhibition of the release of the hormones into the rest of the body. Fair enough? Does that make sense? I only see one yes. That means it's confusing. Yeah, you're right. I didn't mention the rest of the name of the arteries because I don't know if you need to know them or not. Anyways, then you have the nervous system. And the easy way to remember the nervous system is by remembering that posterior is the one responsible for neurons or neurohypophysis. Okay? So again, it says hormones of the posterior pituitary. So immediately from the hypothalamus, you're going to have a nerve ending up at the posterior pituitary and releasing at the posterior pituitary to the rest of the body. Okay? And if you remember back to the type of hormones we talked about, do you know which type of signaling this would be? From a nerve ending into a blood vessel? Excellent. It's neuroendocrine, exactly. So from a nerve ending into the blood vessel and then into the rest of the body, that's known as neuroendocrine. And we have two main hormones being released. Does anyone know them? I mean, it's written right there, but all right. So it's ADH, exactly. It's ADH and oxytocin. Perfect. So the two main pathways, we have the anterior pituitary goes through which system? Is controlled by which pathway? Portal venous, yes, exactly, or portal vascular, exactly. And then you have the posterior pituitary or the neurohypophysis. How is it controlled? Yeah, neuronal. You can call it nervous system, neuronal, or hypothalamo-hypophyseal nervous system, whatever suits you. Perfect. Okay. And then we said sometimes they classify hormones either by local acting or general hormones that act on the rest of the body. Sometimes they classify it based on the chemical structure, which is important to know. So we have steroids, okay? The special thing about steroids is that it's lipophilic. And do you guys know what lipophilic means or why is it important to us? Exactly, it can go through the cell membrane. And then we have the proteins or polypeptides, which is sorry, hydrophilic, which means it does not cross the membrane. And then we have derivative of amino acid tyrosine, which is only the thyroid hormone, and um, and even also the adrenal medullary hormone. Okay, so we have three types. We have steroids, type 1, and we have proteins and polypeptides and derivatives of amino acids. Okay, these are the three types of hormones by structure. Okay, so if the hormone is circulating in the bloodstream, why don't they just act on every single cell? Exactly. Someone said in the chat, it's because of receptors. So, yeah, it's not the same receptor. Exactly. So each hormone has a target cell, a very, very specific cell it acts on. And the reason behind that is that they have specific receptors they bind to. Okay? And it's like a lock and key interaction. If you can see here, this key, whoa, this key, I don't know why I'm not able to scroll. Okay, this key would not fit here, as opposed to this key, it would fit here, right? So it's exactly like that for hormones, where specific hormones will bind to specific receptors, while other hormones cannot bind to the same receptor. That's why we have very specific action sites and target cells. All right. So now the location of the receptor depends on the type of hormone. Okay. And I already mentioned this, right? We said we have hormones that crosses the cell membrane and we have hormones that do not cross the cell membrane. Okay. So we have three different types of location for the receptors. All right. So we have membrane receptors, and as you guessed, it's on the membrane from the name. It's located on the membrane. And can you guess which type of hormones will be acting on this one? I mean, honestly, at this point, it's like Dora. Like it's right in front of you, the answer. Hydrophilic, exactly. So it's peptides and proteins. Um, so because these hormones cannot cross the cell membrane, and then we have cytoplasmic receptors, which are basically located in the cytoplasm. Okay, let me see, where's my cursor? God. Yeah. Which is basically in the cytoplasm. Okay. And these are basically the ones that can diffuse through the cell membrane. And we talked about the steroid hormones. Okay. And finally, we have nuclear receptor, which is basically they're immediately acting on the nucleus. I'm sorry, I'm losing my cursor again. Yeah, so they're immediately acting on the nucleus. And this is basically for a thyroid hormone. So if they ask you which hormone acts immediately on the DNA or directly binds to DNA, immediately answer thyroid hormone. Okay? Which one directly binds to DNA? It's thyroid hormone. There is no other answer. Okay, so three different types. We have membrane receptors, cytoplasmic receptors, and nuclear receptors. Is this clear? Yeah, T3 and T4. Okay, perfect. Do you guys have any questions so far? Or is it clear? Clear? Okay, perfect. I know I ask a lot if it's clear or not, but it's just to make sure. Okay, so how does a hormone work? Okay, so the hormone tries to regulate the cell through something called signal transduction. Okay, And what signal transduction means, it's basically you take the first messenger, which is the hormone. So we call this the first messenger. And we convert it to second messenger, which is basically inside the cell. So let's say, for example, cyclic AMP. And this second messenger is going to basically cause the end result that we need. Okay, let me give you an example. You ate a heavy meal, okay? And this heavy meal is going to cause, for example, an increase in insulin, okay? Now let's say insulin is what? Insulin here is the first messenger, okay? It's going to go in the cell, it's going to cause a signal transduction, and then it's going to increase, for example, tyrosine kinase, which is a secondary messenger. So what does this tyrosine kinase do? It's going to cause, for example, increase in glycogenolysis. Or no, sorry, it's going to cause increase in gluconeogenesis. Glycogen, I cannot say it. Basically, producing glycogen is going to cause increase in glycogen production. Glycogenesis, that's the one. Sorry, guys, it's been like 5 years since first year. Sorry. So glycogen production increases. And how does that happen? Through the second messenger. So we have first messenger producing a second messenger or being transduced to a second messenger. Second messenger produces the end result, which is, for example, one of them is increased glycogen production. And it's not just one action, it's many actions. Okay? So this is just a simple example. All right? So what are the components of signal transduction? We have a receptor, and obviously we have a hormone, and then we have a transducer, which is the G protein, and we have an effector, which is an enzyme, and then we have a second messenger. Okay? This is not an exclusive list, it's just a simplified one. Okay? So the first messenger is the hormone outside cell, and we gave the example of insulin. The second messenger is basically the one that transmits the signal inside the cell. All right, and amplifies it. For example, cyclic AMP, cyclic GMP. By the way, have you guys heard of cyclic AMP and GMP? I'm just assuming you know it. You want me to explain it? I could. Okay, perfect. So I don't need to explain it, right? Okay, perfect. All right. So let's talk more about the role of the second messenger. So the second messenger, we said it transmits, that's number one, and amplifies the signal from the receptor. That's number two. Okay. So for example, let's take cyclic AMP. Cyclic AMP is one of the most common second messengers. Okay. And the way it works is by activating phosphokinase A enzyme. Okay. Phosphokinase A enzyme, cyclic AMP also can also activate some calcium channels. Okay. And finally, it's hydrolyzed into 5-AMP. So here you can see it. This is cyclic AMP. Okay. It's basically can be hydrolyzed, it can activate ion channel, it can donate its, why is it not working? 1 second. Okay, it's back. It can open an ion channel, it can work the enzyme, and finally, it can cause increase in the transcription factors. Okay? So essentially, this is all done by cyclic AMP with a second messenger. And then it can be dissociated into AMP. Okay? And then everything downstream can cause further action. So for example, the phosphokinase A can lead to a lot of phosphorylation in cellular proteins, can open the ions, can cause transcription factors. So basically, the second messenger is activating stuff, and these stuff are activating more stuff. You can think of it this way. Okay? This is the most basic way of thinking about it. Okay, and we're going to talk about this stuff. I'm not just going to say stuff. All right. And we said it's amplification as one of its job for the secondary messenger. What I mean by this is that we have one hormone. Where is it? So we have one hormone binding to one receptor. Okay. This one receptor is going to activate adenyl cyclase, okay, which is going to activate 100 cyclic AMP. This 100 cyclic AMP is going to activate 100 protein kinases, which is going to phosphorylate 10,000 proteins or enzymes, which is going to lead to 1 million products. Okay. Now, why is this important? Okay. The reason I mentioned this is, remember in the very first few slides, I mentioned that we need little amount of hormones to produce a lot of actions. This is exactly why, because we only need one hormone to produce 1 million products, which is a huge number. Okay? Whether that's phosphorylation of enzymes, or transcription of factors, or opening ion channels. Okay? So just remember that. Does that make sense? All right. Um, so let me see hormones and cycling. Yes, exactly. And we're going to talk about it. So this basically summarizes the secondary messengers and their mechanism of actions. And before you freak out and say, do I have to memorize every single one? I'm going to say yes and no. So yes, they're probably going to ask you one of these, and it's going to be something very obvious. Like for example, nitric oxide with cyclic GMP. Maybe it's not obvious now, but it's going to be repeated to you a lot of times, so you're going to remember it. Okay. So I wouldn't say sit down and memorize this table. Just go over it a few times maybe, and then over the lectures, you're going to see it over and over again, which will make you memorize it. Don't worry too much about it right now. Okay. So, um, do you guys have any questions so far? Okay. All right. So, mechanism of actions of hormones. So we're going to talk about some of the mechanism of action of the hormones. Okay. And what I mean by that is the intracellular pathway. Okay. Starting with cyclic AMP. So someone already mentioned how it works. So what happens is that first step is that we have the hormone binding to the receptor. Okay. So the hormone, which is the first messenger, binds to the receptor. And do you guys know what type of receptor this is? Mhm. Exactly. G-coupled protein receptor. Tyrosine kinase is not the right answer. Yeah, I think tyrosine kinase is one for insulin, if I'm mistaken. But yeah, anyways, so this is G-coupled protein receptor, and this is specifically GS protein receptor. So what's going to happen is that the messenger, which is the hormone, is going to bind to the receptor. This binding of the receptor is going to convert GDP, sorry, to GTP, which is the active form. So it's activating it. And this GTP is going to activate adenyl cyclase. Okay. And after it activates it, it goes back to GDP. Now, once adenyl cyclase is activated, it's going to convert ATP into cyclic AMP. Okay. And then cyclic AMP, which is the secondary messenger, what is it going to do? It's going to activate the cyclic AMP dependent kinase, or the protein kinase. That's another way of saying it. Okay. What does the protein kinase do? It just starts phosphorylating proteins. So this is a normal protein plus an ATP molecule is going to lead to a phosphorylated protein. And this phosphorylated protein is either going to lead to cell transcription, ion channel opening, or whatever action it does downstream. Okay, so let me repeat that. Number one, we have the hormone first messenger binding to the membrane, okay, membrane receptor, which causes a change in the shape and causes binding of GTP. Okay, G protein is activated when it has GTP. Okay, and what is it going to do? It's going to activate adenyl cyclase. Okay, now adenyl cyclase, once it's activated, is going to convert ATP to cyclic AMP, which is a secondary messenger. And then cyclic AMP is going to activate what? Protein kinase. Okay, and protein kinase is going to lead to phosphorylation of the molecules. Should I repeat that more slowly, or does that make sense? I want to keep it as simple as possible. Okay, perfect. Are you guys lying to me, or is it that easy? I mean, okay, I'm glad it's easy. I'm glad it's easy. Alhamdulillah. Okay. Then we have another pathway, the PIP calcium signaling pathway. Okay. This one is a little bit more complicated, but it's straightforward, inshallah. So first of all, we have the first messenger, which is a hormone that binds to the receptor, like the last one. And then the same thing happens here. We're going to have binding of GTP, which is going to activate something else now. It's going to activate something called phospholipase C. Okay? Now, in the membrane, we have PIP2. Okay? In the cell membrane, we have something called PIP2. Phospholipase, from its name, is lipase. So it's basically cutting it down. It's cutting down PIP2 into IP3 and DAG. So two molecules, IP3 and DAG. Okay? Now, IP3, which is, we're going to number one here, is going to go down to the endoplasmic reticulum, is going to open ion channels here, and these ion channels is going to lead to the release of calcium. And calcium is going to lead to cellular response. Okay? So IP3, which is one of the products of PIP2, is going to go down, is going to go to the sarcoplasmic reticulum or the endoplasmic reticulum, open the ion channels, the calcium ion channels, and release calcium, increasing the calcium in the cell. Okay? That's number one. Now, DAG, which is number two, is going to go to something called protein kinase C and is going to activate it. Now, what is protein kinase going to do? Protein kinase C, it's like protein kinase A. It's going to lead to phosphorylations, which is going to lead to cellular response. You have two things happening here. IP3, opening calcium channels in the endoplasmic reticulum, increasing the calcium in the cell. And DAG, activate protein kinase C, leading to phosphorylation of different proteins in the cell. Okay? Starting from the beginning, the hormone is going to bind to the receptor. Receptor is going to get activated. Conformational changes. GTP binds to the G protein, activating phospholipase C. Phospholipase C cleaves PIP2 into these two products, IP3 and DAG. IP3 will increase the calcium by opening the endoplasmic reticulum calcium ion channels. And DAG is going to activate protein kinase C, which is going to lead to phosphorylation of proteins, leading to cellular response. Does that make sense? Okay. Now we have also something called calmodulin, okay, which is it's a way basically of amplifying the response, cellular response to all of this, okay? And let me see if I have a picture for it. Oh, that's unfortunate. The way it works, basically, calcium will bind to it, okay? And this calcium will, because they bind to it, it helps with the activation and phosphorylation of the proteins. So it's basically like it increases the action, okay? Why? What does the calcium released by IP3 do? Okay, so calcium, where is it? Yeah, so here it is. So calcium released by IP3. So it's not released by IP3, it's released by the endoplasmic reticulum through IP3. Yeah. What it's going to do, it's going to lead to multiple actions. So let me give you an example. Since you asked, and this is something you don't have to know, okay? Do you guys know how a neurotransmitter is released? Not in detail. Okay. So usually we have some vesicles around the end of the cell. Okay. Sometimes these calcium channel, because they increase the positivity of the cell, they can activate voltage gated channels. And this will lead to basically influx of calcium, further influx of calcium, which is going to lead to release of these neurotransmitters. So this is one example of an action that can happen. Okay. You absolutely don't need to know this, by the way. I'm just giving you just to connect the dots. Does that make sense? Okay, perfect. Now let's move on to steroid hormones. And do you guys remember what we said that's special about steroid hormones? Um, so DNA is not entirely correct. I'll tell you why. Because we said whenever we say binding directly to DNA, we're talking about thyroid hormone, not steroid hormones. Okay? So it's lipid soluble, okay? And it will act eventually on DNA, but it does not directly bind to the DNA. So I don't want you to get confused about it. Okay? So I'll let you know. So first of all, steroid hormones, they're lipophilic, meaning they can cross the cytoplasm. Sorry, they can cross the cell membrane. Okay? Now, because they can cross the cell membrane, this is the steroid hormone, okay? They have the receptors inside the cytosol. So this is the receptor. Okay, let's say R. Okay. Now, this hormone is going to bind to the receptor, and once it binds to the receptor, it's going to diffuse into the nucleus. Okay, this is called the receptor hormone complex. Literally because it's a receptor hormone complex. It's nothing like crazy. Okay, on the DNA, we have something called HRE, okay, which is the hormone response element, okay? And what it means basically, if the receptor hormone complex bind here, it's going to increase the transcription of the hormone. And this way you have more mRNA goes to the cytosol, and you have more protein production, and it's going to lead to effects that we need. Okay, so first, the steroid is going to diffuse. It's going to bind to the receptor. The receptor hormone complex is going to go to the nucleus. It's going to go to the HRE, or the hormone response element. Let me see if it's written. Yeah, it is. Hormone response element. And then it's going to increase the transcription. It's going to lead to transcription of mRNA. mRNA is going to be translated into proteins. Proteins are going to produce effects. Does that make sense? Exactly. Can you repeat one more? Absolutely, I could. Let me just. Okay. So we have steroid. This is the hormone. S is the hormone. Okay. Steroids. Because they're lipophilic, they're going to go through the cell membrane. They're going to diffuse passively. Okay. And now, once we have steroid hormone, they're going to bind to its own receptor. Okay. So this is the first step, diffusing through the membrane. Second step, which is binding to its receptor. Once it binds to the receptor, the whole receptor. So this is the steroid and this is the receptor. Once it binds, it forms something called hormone receptor complex. Okay. This hormone receptor complex, which is this, okay, is going to go into the nucleus. Okay. And it's going to go somewhere on the DNA, which is known as a hormone hormone response element. Okay. It's basically an area on the DNA. Okay. And binding specifically to that area, what it does is it increases transcription of an mRNA. Okay. This increase in transcription of mRNA means that we have more mRNAs to go to the cytoplasm to be translated into proteins. And these proteins are going to produce the biological effects that we want. Okay? Does that make sense? If it doesn't, I'll repeat it again. It's fine. All right. Perfect. Now here's the thing. Forget about this picture. I didn't find a picture for thyroid hormone. The difference between thyroid hormone and the steroids is that thyroid hormones would directly bind to the HRE. So forget about all these steps. They're not there for thyroid. So it's going to go directly into the HRE, binding here, increasing transcription of mRNA, leading to more translation and more biological effects. Does that make sense? So instead of having to form to bind to bind a receptor in the cytoplasm, it goes immediately and binds on the DNA. It's just built different, you know? No, it has a receptor, but the receptor is directly there on the DNA. It's already put there. The receptor will be here, waiting for it to come. All right. So, um, now I found this slide in the second lecture, randomly there. Why is it repressing gene expression? It's not repressing it. It's causing gene expression. Does it say repressing it? Oh, okay, okay. So the statement here, it says that it's already bound, the H already bound to the transcript. Because it's bound to it, the HRE, the receptor, sorry. Wait, let me explain this here so you don't get confused. So this is the DNA. Wow, terrible DNA. Anyways, and we have the thyroid receptors here, okay? These thyroid receptors, when they're not bound to the thyroid hormones, the mRNA is inactivated or the transcript is inactivated. But when the thyroid hormone binds, it gets activated. So what it's trying to say is that before the binding, it's repressed. Yeah, and this is what it says here, results in the activation of the transcription. So a receptor alone with no thyroid inactivates it. Once the thyroid binds, it activates it. Good question, good catch. Exactly. Whoever wrote that statement, exactly. All right. So then we talk about paracrine signaling. So paracrine signaling, from you guys remember, if you guys remember, it's basically cells acting within a close vicinity. Okay. So an example of this would be a cell releasing nitric oxide. Okay, so this is, for example, an endothelial cell. Okay, it has acetylcholine binding to the receptor. We have phospholipase C, and if you remember, phospholipase C has DAG and IP, it releases DAG and IP3, etc., etc., etc. Eventually, you're going to have the release of nitric oxide into the smooth muscles. Okay, which is going to lead to what? It's going to lead to activation of guanylyl cyclase. Okay, causing production of cyclic GMP and activation of protein kinase G, which is leading to decreased calcium and smooth muscle relaxation. Now, you don't need to remember this in DNA. It's just an example of paracrine mode of action. So if they ask you, give me an example of paracrine, what's your answer? Nitric oxide. Exactly. An example of autocrine is prostaglandin. Okay? And I'm not going to talk further about this, but it would happen basically during pregnancy, where the cells are going to be releasing hormones for its own self, so that increases the contraction more and more. All right? Cytokines are exactly. That's also correct. All right. So we're going to have a checkpoint right now. Uh, just like make sure everything is well understood. Do you guys understand everything? Everything is going well? Yeah, that's all of us. All right, so I have a question for you guys here. So type the answer in the chat. Thank you. I'm not the GOAT. Perfect. So do you guys know why the answer is A? Exactly, because it's lipophilic or fat soluble. Exactly. Do you guys want to keep going or do you want like a 5 minute break? Keep going? Okay. Because we have a few more slides. Inshallah, nothing tough. And I'm sorry, it was scheduled for 1 hour, but it's very important to understand rather than just memorize and bombard you guys with like information. So I'm sorry about that. Okay, so we have two major concepts that are very important and is going to be repeated with you in medicine over and over again. It's up and down regulation. Okay. And from the name apply, like from what the name applies, basically, up regulation is basically you're increasing the number of receptor. Where is it? Here it is. So upregulation is increasing the number of receptors compared to normal, while downregulation, you're decreasing the number of receptors compared to normal. Okay? So why do we have upregulation and why do we have downregulation? Does anyone want to take a guess? I mean, yeah, basically your answer is to live. I mean, yeah, obviously, but why? In a way, it's a feedback, but it's not the feedback we talk about in the body. So regulation. Well, you guys are giving very specific answer. I really appreciate it. So let's say for a long time, so the cells don't stop working because of constant secretion. Let me further specify. Okay, so let's say for a long time you have a high level of hormone being produced. Okay, let's say you have insulin being produced really high. Okay, now what's the issue here is that we don't want the cell to be constantly doing glycogenesis. We don't want it to be constantly producing glycogen. So the cell's way of counteracting this, if we have too much insulin all the time, is that it's going to say, you know what? Even though there is a hormone outside, I'm going to take a few of these receptors and put them inside so that the action is less. Okay, and this is what's known as downregulation. Okay, so that even though we have chronically or over a long time high insulin, we don't have the bad effects of insulin all the time. Okay, does that make sense? Okay, I'll repeat it. Let me see if I can get a um trying to get an empty slide, but it's fine. I'll just figure it out. Okay, so let's say for a long time, this is our cell. This is our like a human cell. And these are the receptors. Thank you. Been working them. Now, if we have, let's say, insulin, really high level of insulin all the time for like 2 months. Okay. Now, our body's response, because all this insulin is keeps acting on the cell, and it keeps like producing like glycogen and etc. Okay, it's gonna reach a point where it's gonna be like, okay, fine, you've had enough insulin or you've had enough action from insulin, but I don't want this much anymore, right? It cannot always decrease insulin directly. So what it does is that it takes away some of its receptors into the cell, okay, which is gonna decrease the action of insulin. Okay. So yeah, someone said in the chat, high levels of hormone equals less receptor for it. So does not equal it. It's an end result. So if you have higher level of hormone for a long time, the body's response will be to decrease the receptor. Okay. Now here's the thing. On the flip side, if we have barely any hormone produced for a certain period of time, this cell is going to start questioning its life. It's going to be like, I'm not getting anything, right? So it's going to start putting more receptors. So it increases the likelihood to maximize the production of, to maximize the effect of the hormone. And this is upregulation. Does that make sense? I can repeat it again. You ate that. It doesn't have to do. No. So here's the thing. The body's processes are dynamic. Okay. So even though I'm saying insulin will stay high for a long time, there are ways the body will try to bring down insulin. But I'm saying, let's say insulin stays high for a long time. How does a cell respond? Okay. How does the hormone production changes is a whole different story. Okay. But here we're assuming the hormone production is not changing. Okay. I can give you a clinical example so you guys can stick it to memory and never forget it. Okay? Have you guys heard of a medication that's called beta blockers? Do you guys know what it does? Just generally, you know what it does? So it basically decreases the heart rate and decreases the blood pressure. Okay? And it acts on beta adrenergic receptor. Now, people on beta blockers for a long time, which means basically their receptors are not getting enough hormones, what's going to happen? Upregulation or downregulation? Upregulation, exactly. Now, here's a fun fact. People on beta blockers, it is absolutely deadly for them to stop the drug immediately because they have so many upregulation. They have a lot of receptors like this that if you stop the beta blockers, all the adrenergic molecules will bind and the patient is going to go into hypertensive crisis. Does that make sense? This is something you shouldn't know. I'm just making it fun for you guys. Okay. Yeah, subhanAllah. Indeed, subhanAllah. Okay, then we have hormone control. So this is the one we're talking about. This is a different process for hormone control. It's not upregulation, downregulation of receptor. So we have something called negative feedback mechanism, and we have something called positive feedback mechanism, and we have direct control. Okay. Now, before I move on, I forgot to mention here, it's important to know when we have downregulation of the receptors, they're basically being uh, moved from the cell surface to the intracellular site. Okay. And I already explained it on the cell, but I hope you guys remember that, okay? All right. So, negative feedback mechanism, it's literally what it says. It's basically a negative feedback for one of the hormones. So, if you have too much of one hormone and the body is like, I don't want this much, so what's going to happen? You're going to have a negative feedback on that hormone. And this negative feedback is going to decrease the production of that hormone, okay? And I probably said hormone 17 times in the past few minutes, but bear with me, please. Okay. So let's give an example. Okay. Now, for example, thyroid releases T3 and T4. Okay. And to make sure that the body does not have too much T3 and T4, these T3, T4 will go to the body. It's going to go to the organs. It's going to produce the effects. Okay. And then these T3 and T4 will also act on the pituitary gland and they will also act on the hypothalamus, okay, to produce negative feedback to tell the body, okay, we have enough thyroid hormone, do not produce more of this. Okay, so this is negative feedback. The thyroid hormone goes back to the hypothalamus and to the pituitary gland and tells it, hey, we have enough thyroid hormone, please stop producing. Does that make sense? Okay. Then we have positive feedback. Positive feedback, it's basically when the opposite happens. Okay. So what happens is that we have, for example, where is it? Let me read the example. So, oh yeah. So, for example, at the time of delivery, okay, when we have oxytocin, it has it has positive feedback on the hypothalamus, so it causes more production of oxytocin. So it's basically a positive loop. Okay, so it's increasing its positive production. Okay, and to give you clinical correlation again, what oxytocin does causes contraction of the uterus. Okay, so what that basically, subhanallah, the way we were created, what happened is when the woman delivers, she usually bleeds, right? One way to stop the bleeding is by increasing oxytocin. How do we increase oxytocin? Is by the positive feedback. So this is one way of preventing the woman from bleeding to death. Subhanallah. Allah created it as a positive feedback. You don't need to know this again. It's just clinical coronation for fun. Okay, do these positive and negative feedback make sense? Okay, and then we have direct control, which is, for example, the glucose level controlling the insulin level. It's just a substance in the blood that controls the release of hormone. Another example of this is, for example, the sodium in the blood affects the release of ADH or antidiuretic hormone. And you guys are going to learn this later, so don't focus too much on it. Now, I read a question in the chat that says, what's the difference between negative feedback and wait, let me see, where's the question instead? Yeah, negative feedback and downregulation. Negative feedback is sort of like a hormonal mechanism. Downregulation is a cellular mechanism. Can they happen simultaneously? Absolutely, they can. It's a dynamic process again, and usually I would think of negative feedback as a faster process than downregulation. Downregulation and upregulation are always a chronic process or something that happens over time. Negative feedback, positive feedback can happen within seconds or minutes, or can even sometimes take longer, but you get the point. So does everything make sense so far? Yes, whoever wrote the sentence about the negative and positive feedback, you're right. Okay. Now we have two methods of transporting hormones in the blood. We have, first of all, we have two types of hormones. We know this, the water-soluble hormones, and we have the lipid-soluble hormones. The water-soluble hormones, because they're water-soluble, they can be freely transferred in the plasma. Okay, and then once they reach the target organ, they will diffuse out of the capillaries, and they will go into the target cells. Now, the lipid-soluble hormones, because they're lipid-soluble, they cannot freely diffuse in the plasma, so they bind to plasma proteins. Okay? Binding to plasma protein, so let's say this is a hormone, and this is the plasma protein. The binding of plasma protein does two things. First of all, it acts to transfer, so it's transferring it, and acts as a hormone reservoir. And the third thing, when it's bound to plasma protein, it slows their clearance. Okay? So a protein with the hormone cannot be cleared, but the hormone alone can be cleared. Okay? So it cannot be removed from the body. Okay? Is it clear so far? Because there is one more thing. So when a hormone is bound to a protein, it's inactive. Can you guess how we can activate it? Unbinding, exactly. It's simply unbinding. Once it unbinds, what's going to happen to it? And subhanAllah, it's here. It's going to be like in this form. And then once it goes to the target tissue, it unbinds. And this is why it's going to be able to diffuse into the cell and does all the actions we talked about. Does that make sense? All right. So, just a little about clearance of the hormone. So just few statements, not very important to be honest, but let me see where my cursor is. Yeah, so the rate of the removal of the hormone is basically a metabolic clearance rate. It's literally just saying the same thing, but with fancy words. Um, and this is like a formula for it, not very important. Just know that the way it's cleared from the plasma, the way hormones are cleared by one of these four things. So, number one, it's by being destroyed in the tissue. Number two is by binding into the tissue and ultimately, again, destroyed into the tissue. And number three, it's excreted into the liver, by the liver into bile, which is going to go to the poop. And number four, excreted into the kidney by urine. Okay, so one of these four ways is how the hormone basically is cleared. Because nothing just disappears, there is a process, right? So someone said thyroid bind to plasma proteins, so less clearance, inactive until unbind. Exactly. So it will have less clearance because thyroid hormones are bound to plasma proteins. They're going to have less clearance when they're bound, and they're going to be inactive until they unbind. Perfect. Now we're going to talk a little bit about methods that are used for hormonal assays. Okay, so an assay is basically a way to detect and measure the concentration of hormones. Okay, so for example, when you go do a blood test, you want to see your insulin level or you want to see your cortisol level. All of this is basically done through an assay. Okay, and you do it either through a blood sample, a urine sample, or saliva. You can also do it through the CSF, but that's something else. So let's focus on these. Okay. So again, an essay, I wrote the definition again. So we have something called biological assay. So we have different types of assays. We have biological and we have immunological and we have chemical. Okay. So the biological assay, it measures not directly the hormone, but the biological effect of the hormone. Okay. What do I mean by this? So instead of directly measuring insulin, we measure how it changes the blood glucose. Okay? So instead of measuring the actual concentration of insulin, a biological assay will go ahead and measure its action or biological action, which is decreasing the glucose. Does that make sense? Okay. And this is what hormone units are. It basically, when we say, for example, 100 units of insulin, okay, it's not really showing the mass of insulin, but rather the effectiveness or biological effectiveness of insulin. Okay, so just keep that in the back of your mind. Just understand the concept, don't memorize. Is it clear? Okay. And then we have immunological assays. Okay, so these immunological assays, they use antigen antibody reaction. Okay, and you guys know from before that antigen will bind to antibodies. So scientists decided to take advantage of this, that if they know the antigen and they know its structure, they can find an antibody they can bind to and specifically measure the hormone. Okay. Now, before I move on, to make it clear for you guys, the reason we use biological assay, or people used biological assay, because scientists did not really know the chemical structure or composition of certain things. So they didn't know how to make an antigen antibody complex. So they decided to measure the end result, which is, for example, the blood glucose. That's the reason why this happened in the first place. Does that make sense? But then things advanced and they were able to find specifically antibodies for the antigens, which are hormones, okay? And they have a very precise detection of the hormone, okay? So the hormones here essentially, they're also the antigens, and then they produce antibodies towards that. This is an antibody. Okay. And we have two main ones. We have the radioimmunoassay and we have ELISA. Okay? So let's talk about radioimmunoassay first. Radioimmunoassay, the way it works is that it works by competitive binding. Okay, so how does that work? We have two hormones. We take a sample from the blood of the patient, okay, and we produce, so we have a normal hormone that's in the blood, and we have a radioactive hormone, okay, and we have an antibody. So this is the antibody. So the concept of this is that we have an antibody here, right? And the antibody will bind to the antigen, okay? If we have more hormone, regular hormone in the body, it will bind more to the antibody. Sorry, if we have more hormone, which is the antigen, it will bind to the antibody more. So less of the radioactive hormone will bind to this antibody. So there will be less radioactivity. Okay? So more normal body hormone equals less radioactive. Does that make sense? It's counterintuitive, I know. One more time, please. Okay, no problem. So we have antibodies in the body. Do it again. Okay. So we have antibodies, not in the body. We have in the assay. Okay, we have antibodies. Okay, we know this antibody specifically will bind to the hormone. Okay, and we have produced two types of hormones. One, which is the endogenous hormone, or the one that the body produces. Let's call it endogenous. And two, the one we produced in the lab, okay, that's radioactive, or also known as a tracer. Essentially, these two are competing against each other for the same binding sites. Okay, so scenario number one, we have a lot of endogenous hormone, so more of it will bind to the antibody. Okay, so less of the radioactive will bind to it, to the same antibody. Does that make sense? Okay. Scenario number two, we have less of the endogenous. So what's going to happen? We're going to have more of the tracer or the radioactive. So it's going to be shiny on the microscope. Does that make sense? Okay, so here's basically a summary. High radioactive binding, it means we have low endogenous hormone. Low radioactive binding means we have high endogenous hormone. And I'm sorry if you hear bird noises, my bird is going crazy. My apologies. Okay. Does anyone have any question? All right. Then we have ELISA or enzyme link immunosorbent assay. Okay. And basically, the way this works is that, first of all, we have a plate with antibody number one. Okay, focus with me because this is going to get a little bit complicated. Okay, so in our patient's blood, we're going to have the hormone. Okay, this hormone is going to go to the plate and bind antibody one. So this is the plate, and this is antibody one. And this is the patient's hormone. It's going to bind to antibody one. Okay, after this, we add antibody two. Okay, which is going to bind at a different spot from antibody one. Okay, now after antibody 2 binds, we add a different antibody, okay, which is antibody 3 with an enzyme. Okay, why do we do that? Because antibody 2 is solely like an anchor for antibody 3, and antibody 3 has an enzyme which is going to cause activation of molecules, which is going to lead to fluorescence. And fluorescence is basically the way we quantify the hormone. Okay, so Our hormone is going to come from the blood, go to the plate, it's going to bind to the plate's antibody, which is antibody number one. And then we have the antibody number two, which is kind of like an anchor to antibody number three. Antibody number three has an enzyme attached to it, which is going to cause fluorescence, activation of the fluorescence, okay, colored product, which can be directly quantified. And this is how we know how much hormone do we have. So more color equals more hormone. Does that make sense? Okay. Now here's the trick. ELISA, if we have more hormone, it means more color, it means stronger signal. So basically more hormone, more color. In radio immunoassay, if we have more color, we have less hormone. Does that make sense? I don't want you guys to get tricked in the exam. That's why I'm explaining it. Feel like you guys got lost. Yeah, I can repeat ELISA. Do you want me to repeat the ELISA, like the concept or the last statement? The concept. Okay, no problem. So, first of all, start from the beginning. Let's say we're at the lab, okay? This is a plate, okay, that we get from the lab, and we coat it with antibodies, which is, let's call antibody 1 for the sake of easiness, specific to a hormone that we want to measure. Okay, so let's say this antibody is specific to this hormone. Now we take a sample from the patient, okay, which has the hormone, put it into the plate, and guess what? Because it's specific to it, the hormone is going to bind to the antibody. So far, we only have one antibody and one hormone. Okay? Now, after that, we add another antibody that's going to bind to the hormone from a different place because it cannot bind from here. It's going to have to bind from here, simply. Okay? Now, this antibody's sole purpose is to create an area for antibody 3 to bind to it. Now, why do we need antibody 3? Because it has an enzyme. The enzyme is going to convert the substrate. It's going to convert the substrate to a colored product. And more colored product, it means that we have more hormone and it's quantifiable. Why do we want to convert a substrate to a colored product? So we're able to quantify. We're able to tell how much hormone there is. Okay? Does that make sense? So the only thing that's coming from the patient is the hormone. The rest is part of the assay itself. And enzyme 3 basically is a way of telling us that hormone is there, okay? Because let's take on the other hand, the patient has no hormone in his blood, or the hormone that we're trying to measure, there is no hormone. So there will be no hormone here. So even when you add antibody 2 and antibody 3, they're not going to bind to anything. So there will be no color or less color. So less color means what? Less hormone. Does that make sense? Okay, no worries. Okay, final concept of the lecture, and then we're done. All right. So now there are some kinetics when it comes to binding of the hormone, okay, and hormone binding to the receptor. Okay, so a biological response will depend on how many receptors are bound, which makes sense. The more receptors that are bound, the more biological response we're going to have. And this is going to depend on what? The hormone concentration, number of receptors, and the affinity of binding. Okay? So how well does the hormone bind to the receptor? Okay? And this is well represented in a graph that looks like this. Okay? So, First of all, let's take it step by step. So the x-axis of this graph represents the amount of hormone that's bound. Okay? The y-axis is presenting the bound hormone divided by the free hormone. So the ratio of bound hormone to the free hormone. Okay? And then the slope of this graph is representing the negative of the affinity. Or you can just say the affinity for ease of sickness, but it's the negative of the affinity. Okay? So the more steep it is, the higher the affinity, the stronger the binding. And finally, the x-intercept, which is the point at which the x-axis, the y-axis is at 0, is the number of receptors. Okay? These are just things that you need to like remember. Like I cannot explain it. It's just something you have to remember. But what you need to remember from here is a couple of things. The higher the affinity of binding, the more tightly the hormone will bind, and the more the biological response is. That's the first thing. The second thing is that the slope represents how high is the affinity. So if the graph is like this, that means we have less slope. So what happens to the affinity of binding? Less affinity, exactly. It goes down, exactly. You guys get it. All right. So some hormones doesn't have a straightforward curve. It does not have like this type of curve. It has like a different type of curve, like the one we're looking at. What this means is that we have two different type of receptor. One with high affinity and one with lower affinity. Okay. And as you guess, this one will have which high or low affinity? Exactly. This one has high affinity and this one will have low affinity. So what will happen is that this is, for example, for insulin. Initially, insulin is going to go to the higher affinity because it has higher affinity for it. It's going to bind, all the way to this point where it's basically becoming almost saturated, right? And then it's going to start binding to the other type of receptors until it's fully saturated for both receptors. Does that make sense? Um, no. So you guys have mole? Like, are you taking this currently in mole or last semester? Because I don't want to confuse you. Because if you're taking it this semester, uh, I can teach you. So we have something called KA and we have something called KD. I think you're talking about KD or KM. And what KD or KM, let me explain on the other graph. KD is, where is my cursor here? KD is the amount at which 50% of the receptors are fully bound. Okay? A higher KD equals lower affinity. So this is what you're referring to, right? This is something completely different. We're talking about Ka, which is the affinity. Kd is the amount at which 50% of the receptor are bound. And let me explain it to you. So let me draw a small graph. So this is the graph, right? So when we say higher Kd, I think Km and Kd are the same thing, if I'm not mistaken. I don't remember, to be honest, but I think they are the same concept at least. So a higher Km, it means basically, let's say Kd, the point at which 50% of the receptors are bound. So this versus this. Okay, so which one is going to have higher KD? Let me number them, one or two. Higher KD. 2, exactly. Why? Higher because it's basically the point at which 50% is going to be here, which is basically higher in number versus here. But which one has higher Ka, which is the affinity, which is the slope? 1, exactly. Now, out of this lecture, what I want you to remember is the Ka. So don't get confused, but you asked me, so I'm trying to explain to you. So what I want you to remember from this lecture is Ka, which is the affinity, okay? Yeah, so Kd or Km are the opposite affinity. You're right. So just remember Ka, the higher the steeper the slope, the higher the affinity. The lower the slope, the lesser the affinity. Okay? So is the equation important? I don't think you need to memorize the equation. I wouldn't, honestly. No worries. Okay, can you guys answer this question? Are you sure it's A? What do you guys think? Is it A or is it B? Are you sure it's A? Yeah, it's A. I'm just tricking you guys. It is definitely A. So basically, as you can see, um, the slope. Wait, let me see where my cursor is. I'm sorry. So this one has a steeper slope, so the Ka is higher. So the affinity is higher. This one has a lower slope, so the Ka is lower, so less affinity. All right, you guys are doing great. So I put a few practice questions. So I'm not going to show it, so you guys can solve it later on your own. Um, all right. Um, this is the feedback form. You guys have to scan it. And thank you guys for tuning in. Before I end this lecture, I want to ask you if you have any concept that you wrote down or anything that you feel like you're not good with that you want to revise.